Okay, so just looking at a review here, just to remind you, we've got the autonomic nervous system, we've got the somatic nervous system, that was chapter 11. We've then got the endocrine system, chapter 6 and 21, or at least parts of that. And then we got into muscle. So I'm going to touch on the autonomic nervous system and a little bit of somatic, kind of the differentiating between those three branches of the peripheral nervous system. I'm going to touch a little bit on the endocrine system, two specific topics that I got a few questions on that I will brush up or kind of review. And then I'll look at muscle. So particularly with muscle, I'm going to talk about the Crossbridge Cycling, as well as excitation, contraction, coupling. Those are the really big topics from muscle physiology that were in that recording that you had as independent study. So I'm going to touch on those topics in general. If you've got questions related to those topics, feel free to raise those as we go along. If you have other topics, I'll try to carve a few minutes at the end. You can just drop those in the chat as we go along as well. Okay, so let's start off with a general overview of the three branches of the peripheral nervous system that we talked about here. Okay, so we're looking at the peripheral nervous system, autonomic and somatic. And we're going to divide that into three main parts here. So we've got the somatic nervous system. All right, then we've got the autonomic. Here in brackets, I'm going to put voluntary. We can control this branch. Involuntary, we cannot control the autonomic. Okay. So these are two main divisions. And then going further within the autonomic, we've also got the the sympathetic. So here I'm going to put sympathetic. And then we've got the parasympathetic. Okay. So let's start out with a cross-section of these three regions. So we've got the sympathetic cross-section, we've got the parasympathetic down here, got the somatic down here. And we'll compare different features of each of these. So we know we have our dorsal horn, lateral horn, ventral horn scenario going on here. Okay. All right. And so we know that fibers in the sympathetic arise from T1 to L2. So here we're starting from T1 to L2. Here we're looking at the cranial nerves 3, 7, 9, 10, and our sacral nerves S2 to S4, okay? The fibers themselves arise from the lateral horn. So really important, lateral horn is where the fibers here arise from, and then they leave via the ventral root and travel out into that nerve, all right? So we have the lateral horn and then emerging into our nerve. The sympathetic nervous system, here we have an entirely different name. So we'll talk about the name coming up in a minute, but the fibers arise from the ventral horn and then emerge into that nerve. Okay, so kind of one main difference there. Let's look at the nerves that are involved here. So in the autonomic nervous system, we have preganglionic and postganglionic fibers. These are our autonomic fibers. So we have a short preganglionic neuron for the sympathetic. We've got a long preganglionic neuron for our parasympathetic. And then we've got a long postganglionic neuron here and a short postganglionic neuron for the parasympathetic. Okay, and so let's talk about the sympathetic first, and then we'll jump down and talk about parasympathetic. So this is our preganglionic fiber. Okay, we know that it emerges as a myelinated white ramus fiber. Before it gets to the ganglia, this is our autonomic ganglia. And then it's going to re-emerge from the autonomic ganglia back into the nerve as an unmyelinated gray ramus fiber. All right, so at this point, it is an unmyelinated or a gray ramus fiber. Okay, we also know that there are 3 options for this ganglia. So one is a ganglia that is a part of the sympathetic chain, especially, all right, so unique to the sympathetic system is what I'm trying to say. Unique to the sympathetic system, we have 3 routes of communication of these preganglionic to postganglionic neurons. Sorry, let's label this one out here. So this is a postganglionic neuron. So the 3 routes of communication is 1, a ganglia that is a part of the sympathetic chain. Oops, kind of writing right over that word there. 2 is that is a ganglia that is a collateral ganglia, not a part of the sympathetic chain. And then 3 is that we synapse onto the adrenal chromaffin cell, the adrenal medulla chromaffin cell. And this was really responsible for giving us that diffuse response, right? When we synapse onto an adrenal chromaffin cell that dumps our neurotransmitter into the blood, it becomes a hormone. And specifically this 3rd route gives us that rapid diffuse response. Let's talk more about the parasympathetic, and then we'll speak about receptors and neurotransmitters. So parasympathetic system, pretty straightforward. We've got 1 preganglionic neuron here. It's a longer preganglionic neuron. We've got a shorter postganglionic neuron. Okay, we talk about where these fibers arise from. So basically brainstem and sacral spinal cord. And then we don't have a sympathetic chain. So there's no sympathetic chain in the parasympathetic system. There are no similar structures or synonymous structures, but we do have an autonomic ganglia that is way out here, very close to the effector tissue. And that is where we see that synapse of these 2 neurons. So we have an autonomic ganglia here. And then we get very short distance to the effector tissue. Let's talk about the effector tissues and the junction here. So this is a neuron to neuron synapse, right? And I'll talk about the receptors coming up a bit, but it's different from the neuroeffector junction. So the neuroeffector junction had these nerve endings. So we've got these nerve endings of this postganglionic neuron. And along these nerve endings, we have the varicosities, right? So this is one of the unique locations of these swellings that are the varicosities that have that neurotransmitter stored inside, okay? And so this region is called the neuroeffector junction. Okay. And then the tissues here were examples: cardiac muscle, smooth muscle. So I'll just write them here. These apply for both. So cardiac muscle, smooth muscle, glands, adipose. Okay. All right, let's talk about the neurotransmitters and the receptors, and then we'll get on and look at the somatic nervous system. So the neurotransmitters from our preganglionic neuron is always acetylcholine. So acetylcholine is being released from the preganglionic neurons, always, okay? The neurotransmitter being released from the postganglionic neuron on the parasympathetic side is also acetylcholine. So we also have acetylcholine being released here, but in the sympathetic side, it's norepinephrine or epinephrine. Okay, really important difference here at the neuroeffector junction on the sympathetic side versus parasympathetic side. Okay, the receptors that we see on the postganglionic neuron is obviously going to be cholinergic. So it's acetylcholine. It'll bind to a cholinergic receptor, but it's specifically a nicotinic cholinergic receptor. And I'm kind of out of space here, but I'll just scribble it in, a nicotinic cholinergic. Okay, this is a fast receptor. This is an ionotropic receptor. So it's going to result in an immediate response or depolarization of our second neuron. Likewise, down here on our postganglionic neuron, we also have a nicotinic cholinergic receptor. Same deal, fast receptor, ionotropic, immediate response. At the neuroeffector junction, we have different receptors. So on the sympathetic side, we have adrenergic receptors. All right, we have adrenergic receptors on our effector tissues. This is the D, adrenergic. And these were several different examples that we talked about. So we had alpha 1, alpha 2, we had beta 1, beta 2, and beta 3. Okay, so we'll talk more about the differences with these, but these are on the sympathetic division in terms of these effector tissues. And so they're going to be different types depending on exactly where we are. Cardiac muscle will have beta 1, et cetera, et cetera. We'll talk about this coming up. And then on the parasympathetic side, we have muscarinic cholinergic. So it's also a cholinergic receptor, but here it is a muscarinic. This should be an A, excuse me, muscarinic. This should say C. Let's do that again. Scribbling at this point. Okay, so this is a muscarinic cholinergic receptor. Okay, all right. And then although I'm not mentioning it for each of these, the adrenal clomiphene cell has nicotinic cholinergic receptors. And then that's also going to be applicable in terms of this autonomic ganglia. Even if it's not a neuron, it's a cell, it still has that same means of communication. So on our adrenal clomiphene cell, we will likewise see nicotinic cholinergic receptors because remember, they're acting just like postganglionic neurons in terms of transmitting that signal. Let's move down and look at the somatic nervous system. So here we have one neuron, major difference. We have one single continuous neuron that is synapsing all the way. Okay, here we do have a discrete axon terminal. We don't have what's going on here with the varicosities. And this is called the neuroeffector junction. This is unique because it's only synapsing to skeletal muscle. All right, so this is skeletal muscle. In other words, the muscle that we can move and make voluntary decisions about. And this is called the neuromuscular junction. Okay, and I'm not going to go into the details of the synaptic communication, that transmission, but be familiar with that. Be comfortable with those steps. When the action potential arrives at the axon terminal, the calcium channels, the exocytosis, be familiar with those steps, although I'm not going to go over them in a great detail here. Okay, the structure here, this is called a motor neuron. All right, here we only have one neurotransmitter. It is always acetylcholine, nothing else. One neuron, one neurotransmitter, and we only have one type of receptor. It is always a nicotinic cholinergic receptor. Okay. All right. And then we talked about some other features involved here, such as the motor unit. So a motor unit is a single motor neuron and all the different fibers that it is innervating. So think about, make sure you understand that concept, that a single fiber can innervate many different, or sorry, a single neuron can innervate many different fibers, but a single fiber only receives innervation from one neuron. All right. Any questions here? Any questions on this bit? All right. What we'll talk about now, and again, I'm saving these drawings just in case you missed anything. We'll move on to speak about. Sorry?
Wait, did you? I'm sorry. Is the drawing gone? I wanted to take a screenshot.
Oh, let's see. There we go. Sorry.
Thank you. This will just be a moment.
Okay. All right. Any other questions before we move on from this part? What we'll touch on now are just some of the receptors and kind of getting the mechanisms of those receptors clear. So let's review the adrenergic receptors, and then we'll move on to some other units, some other chapters. So we've got the adrenergic receptors. And I apologize for my terrible handwriting. And so let's just look at just kind of classifying our receptors here, really important. So we have the name of the receptor, we'll do the location, we'll do its affinity, what it prefers, we'll do its second messenger, and then we'll do its effector tissue, its effect on the tissue. So we have the alpha-1 receptors, we had alpha-2, beta-1, beta-2, and beta-3. Okay. So we'll start with, let's actually label this out here. So we've got the receptor, we've got the location, we've got its affinity, its second messenger, and what it does to that second messenger. And then we've got the effector tissue, okay? Specifically the effector tissue response, right? Now remember, these are metabotropic receptors, so really important, I'll write that here in brackets. They're gonna be coupled to G protein, so these are metabotropic receptors. And these are adrenergic, so they only bind norepinephrine or epinephrine, okay? Let's start with alpha-1. So alpha-1, we said, is on most tissues. It's the most abundant, so it's the most abundant adrenergic receptor. And it's usually on vascular smooth muscle. Okay, it has a preference for norepinephrine over epinephrine, much like alpha-2, we'll talk about in a minute, but it prefers norepinephrine, and it is going to increase its second messenger, which are IP3 and DAG. IP3 and diacylglycerol DAG, okay? This is going to be coupled to a G stimulatory protein, hence increase its second messenger. The effect on its tissue will be excitatory. In other words, contraction, just excitement of that tissue. In brackets here, we'll put contraction. Okay, let's talk about alpha-2. Alpha-2 is likewise, it's also very abundant, also found in vascular smooth muscle, so same general responses there. It also has an affinity for norepinephrine over epinephrine. It is going to have a different second messenger and have a different response. So it is going to decrease its second messenger, which is cyclic AMP. And that is because it is coupled to a G inhibitory protein. Okay, so it's going to decrease its second messenger. It's coupled to a G inhibitory protein, but that still results in an excitatory response on the tissues. In other words, contraction. Okay, let's talk about our beta receptors. So we got beta 1, beta 2, beta 3. Beta 1 is found on the heart and kidneys. It has an equal affinity for norepinephrine and epinephrine, so it doesn't really have a preference. It is going to increase its second messenger, which is cyclic AMP as well, and that's because it's coupled to a G stimulatory protein that will result in excitation or contraction to the tissues. Okay, beta 2 is found on the lungs and uterus. It has a preference for epinephrine, so very unique in this regard. It prefers epinephrine over norepinephrine. And then it also will increase its second messenger, cyclic AMP, also coupled to a G stimulatory protein. But this actually generates, because of the unique locations here, this generates a different response, same mechanism, different response because of a different tissue location. So here it's going to be inhibitory to the tissue. In other words, it's going to cause relaxation of that muscle. Okay. And then lastly, we've got beta 3, only found on adipose cells, fat cells, or fat tissue, also having an equal affinity, just like our beta 1, so norepinephrine equal to epinephrine, no real preference there. Also having an effect where it increases its cyclic AMP, second messenger, coupled to a G stimulatory protein, and generates an excitatory response at the tissue level on those adipose cells. Okay. Actually, we're not going to put contraction here. This is not muscle, this is fat, right? Okay, but it's still an excitatory response. Okay, so this is kind of just a general overview of organizing our receptors. Make sure you understand which have a varying affinity, which prefers nothing, which prefers norepinephrine or epinephrine, their second messenger, their effector tissue, and so on. Okay, any questions on this bit, the receptors? Okay. So that's kind of it as far as the somatic and autonomic. Make sure you go over the toxins. I don't have time to really go into the details and kind of reteach that, but go over your toxins, latroxin, cotoxin, what they're doing. Curare was the third one, and then kind of what they're going to cause in terms of a clinical presentation. Is it fatigue, spasticity, rigidity? What type of response those toxins will bring about? So just brush up on your toxins. Okay, let's keep going here. Also in the somatic nervous system, I think we touched on the end plate potential. I didn't speak about it too much in the mechanism, but be familiar with this process or similar steps in terms of that synaptic transmission, but have an understanding of what's happening here. So it is always excitatory. The end plate potential is the name of the depolarizing event on muscle. And then the motor end plate is the unique term that we call this folded membrane of skeletal muscle. Okay, and this kind of picks up the events that I'm going to touch on right now as far as excitation, contraction, coupling. So the events of excitation, contraction, coupling are picking up from this end plate potential. What happens on the muscle after it's excited by these nicotinic cholinergic receptors, right? How do we turn that into the muscle contracting from that motor neuron releasing acetylcholine? So also make sure you're making that connection between the neuromuscular junction and then how that muscle actually brings about contraction. Okay. I'm going to jump over to the endocrine system here. We'll talk about some of the important parts of the endocrine system, and please feel free to raise questions if you have them. But this drawing is probably the most important takeaway. So have a good understanding of these axes, right? So what's happening with the hypothalamus, how those hormones, these are called releasing hormones or trophic hormones. These are going to be released into the portal circulation. That blood then goes to the anterior pituitary, right? Remember, anytime blood goes from one capillary bed to another capillary bed without going back to the heart, that is called a portal circulation. And so that is how the blood that leaves the hypothalamus with those hormones then goes to the anterior pituitary and drops those hormones off. Okay, those hormones will then instruct the anterior pituitary to release another hormone, a secondary trophic hormone. So those can be any of these groups here: prolactin, TSH, ACTH, growth hormone, etc. And then those trophic hormones from the anterior pituitary are then going to instruct the endocrine glands. So we have the distant endocrine glands, which can be breast, which can be thyroid gland, adrenal, cortex specifically, so not the medulla, only the cortex, other cells in the body, as well as the testes and ovaries. And then those cells, depending on the instruction coming down from anterior pituitary, will release their secretions, right? So the breast will have milk production under the influence of prolactin. This is different from oxytocin. Oxytocin is going to be for milk ejection, okay? We then have TSH resulting in T3, T4. We spent a lot more time on this axis, so make sure you have a good understanding of the details here. The fact that T4 is involved in the negative feedback loop. So T4 will go back, feed back up to the hypothalamus to shut off TRH release, or we can have a short feedback loop where TSH goes up to the hypothalamus and shuts off TRH release, okay? We also spoke a little bit more about the cortisol axes. So these two axes we spent a lot of time on, more so the thyroid in terms of that mechanism there. So have a deeper understanding of these two. And then the general flow of GRH, the general flow of GNRH. Sorry, GHRH and GNRH, okay? Any questions from this bit? Okay, another important topic from the endocrine system was the mechanism of thyroid hormone release. So really important that we grasp this understanding how TSH coming from the anterior pituitary binds to this metabotropic receptor, brings about this dissociation, activates the second messenger, generates this protein kinase, phosphorylates this protein that causes the engulfing of these vacuoles. And these vacuoles have thyroglobulin along with residues. If it's got 3 residues, it's T3. If it's got 4 residues, it's T4. And together, this is what we call thyroid hormone. So thyroglobulin is typically stored over here in the colloid, and we need iodide to come in from our diet in order to activate this to be absorbed or engulfed and to generate thyroid hormone, all right? This we call the follicular cell. This entire membrane that's lining the follicle, we call the follicular membrane, and that is how, this is lining those follicles that are inside the thyroid gland, and that is how we can generate or release T3 and T4 into the blood. Remember, T3 and T4 is going to be involved in metabolism, so increasing the rate of the sodium-potassium pump, increasing the metabolic activity, increasing the oxidative phosphorylation in the mitochondria, increasing the number and size of mitochondria, all of those good stuff, all those good things that come along with an increase in metabolic activity, whereas T4 can be converted to T3 peripherally if we need it to, but it can also have that negative feedback element. So it can go back up to the hypothalamus, shut off the whole axis by shutting off TRH, which is decreasing TSH levels and eventually decreasing the thyroid hormone levels. Okay. Any questions from this? So important things from the endocrine system, thyroid hormone specifically, that entire chart with all the various axes as well. And then we got into the fed and fasted states. So make sure you have a good understanding of the general differences here and then the details. What types of events are being associated with the fed state, right? If we're in the fed state, we've got insulin as our main hormone. This is where we have the building up of nutrients. We're going to use what's important. We're going to start building everything up. We're going to have glycogenesis, making glycogen. We're going to have making up of triglycerides. We're going to have storing up of amino acids into proteins. And so that general understanding that this is an anabolic set of events, right? We're building things up for later use under the influence of the fed state. The fasted state was the opposite, the main hormone being glucagon. We're breaking things down here. We're trying to liberate energy until we can get to our next meal. We're breaking down muscle to find stores of glycogen or of amino acids if we need to. We're using beta oxidation to generate fatty acids from triglycerides. So actually, let's go here. So we're kind of moving in the opposite direction to break those things down to liberate energy. Also remember that the brain is kind of spoiled in that it only likes glucose. So we're going to have to either use ketones, which can somewhat be converted to that form of energy, or spare that glucose for the brain, kind of reserve it for the brain and use it exclusively for the brain. Whereas other tissues can use amino acids, they can use fatty acids, they can use ketones as well. Okay, and I'm not going to go into much more detail than that, but have an understanding of those two major states, fed versus fasted, as well as the hormones involved. So insulin coming from the beta cells, being a hormone of the fed state, anabolic events, glucagon coming from the alpha cells, being a hormone of the fasted state, catabolic events. Okay. All right, any questions from the endocrine system? Any questions from the endocrine system? See how we're doing with time. Okay. Lastly here, I'm going to move on to muscle phys and talk about some important things that we encountered in muscle phys. One was the staining patterns here. So the bands and zones, have a good understanding of these. What is forming the I band? Actin or thin filaments. What is forming the A band? This is an overlap. So we have both thin and thick filaments, both myosin and actin. What is forming the Z line? This is the anchoring or the attachment between the actin filaments where they're connected to each other. What is forming the M line? This is the anchoring or attachment between the thick filaments or their connection right down the center. What is forming the H zone? This is the area of only thick filaments, no overlap. So only myosin, no overlap. Also, the confines of the Z line. So the definition of a sarcomere is Z line. It's a Z line. That's going to be important as well. Although I'm not going to go over the general anatomy, so the myofibril, the epimysium, endomysium, go over that, but also have an understanding of the staining patterns. So the A band, the different zones, and what is actually making that up. Other important thing here that I'm going to spend a little bit more time going over was crossbridge cycling. And so crossbridge cycling is how we can use calcium to regulate the process of actin and myosin interaction. And this is how we shorten the sarcomere, which eventually leads to muscle shortening. So we start out with the binding of myosin to actin. So myosin reaches up, grabs onto actin. This is because the myosin head, this thick filament head, is in its high energy form, right? It's got a phosphate bound onto it. That phosphate has been hydrolyzed. So this organic phosphate is ready to give the energy. And so it's going to reach up, bind to actin. It's really attracted to actin in this state. And so that first interaction is made. Then it releases that phosphate off. So the releasing of that phosphate off is in coordination with the dragging of actin. So myosin is reaching up, grabbing onto actin and dragging it towards the M line, towards the center of the sarcomere, which is essentially bringing the sarcomere together, shortening the sarcomere, bringing those Z lines closer to each other. That will then result in the power stroke. So that dragging action is called the power stroke as actin gets pulled towards the middle of the sarcomere. We then see the ADP come off. So the phosphate came off first, then we have the power stroke, then the ADP comes off, and then we have what's called that rigor state. Rigor state is when myosin is in its low energy form. It doesn't have the ADP or the phosphate bound to it. It's still attracted to actin because it doesn't have a new ATP bound yet. And so it's going to stay tightly bound to actin until a new ATP comes in and releases it back to its other form, right? Releases it back to its relaxed state. So this is called rigor mortis. This is why when someone dies or they expire, unless we have ATP in the body, the muscles stay very tightly contracted in the position that they were in at the time of death because of this phenomenon called rigor. We then see a new ATP bind in, and then we can release that myosin from actin because of the presence of that new ATP. And then we get back to it being hydrolyzed and back into its high energy form. Okay, so just remember the general principles here. When ATP is hydrolyzed, the myosin head is now attracted. It's in its high energy form and it is attracted to actin. When the ATP is on there, and it is not hydrolyzed, myosin does not want to bind to actin. That's why we need that new ATP to come in to break that interaction between myosin and actin. And this is happening, we're kind of zooming in on this happening in one cycle, but this is happening over and over and over and over again until we shorten the sarcomere. So it's reaching up, dragging actin, breaking contact, reaching up, dragging actin, breaking contact. And that is happening gradually, 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 gradually until we shorten the sarcomere to its shortest configuration where the muscle will be in its most contracted state. All right, so that was crossbridge cycling. And if you haven't already, check out this video. It's really good at kind of breaking this down, simplifying this. The other thing I'm going to review here quickly was excitation, contraction, coupling. So that was how we can match the events of what I just explained to the presence of calcium. So just reviewing here, calcium is what is regulating this whole thing. If there is no calcium present, then the myosin binding sites are covered up. Remember, when calcium comes in, calcium are these green dots here. They're typically being stored in the SR, the sarcoplasmic reticulum. And when they get released, they will then flood the cytosol where these actin filaments are, right on the myofibril. They'll bind on to troponin. And because troponin is a complex of 3 proteins, they will drag that second complex, which has the tropomyosin bound to it. And so when they drag that tropomyosin off, they uncover these black dots, which are the myosin binding sites. They expose those myosin binding sites such that myosin can now reach up and make contact via those binding sites. So if there's no calcium present, as we're seeing here, then those binding sites are covered up and myosin cannot make contact with actin. So that is how calcium can turn this on and off, right? So let's start out here with the events at the neuromuscular junction. We've got acetylcholine coming down our motor neuron. It's crossing our junction. It's binding to these nicotinic cholinergic receptors. That is resulting in the end plate potential. After we have that end plate potential, that electrical energy, that depolarizing event travels right throughout the muscle via these T-tubules, the transverse tubules. So these are these invaginations that are running deep throughout the muscle that can spread that excitatory event very quickly and can result in the excitation of the entire muscle. It is also coupling the release of calcium from the SR. So because of the ryanodine receptors that are on the membrane here, when the electrical activity gets into this space, it is going to signal these ryanodine receptors, which will signal the calcium channels, the DHP receptors, and that will allow for calcium to go from the SR into the cytosol itself. Calcium can bind onto this troponin complex, so the pink little dot here, the pink little complex here. Calcium binding onto that complex will have the effect of dragging those tropomyosin, so the thread-like proteins here, the purple ones, those get dragged off of the binding sites when calcium comes in. That will allow myosin to now make full contact at those binding sites, start that crossbridge cycling that we just described, shortening the sarcomere, and then that will generate that muscle contraction. In order to cease that muscle contraction, we have to let that calcium leave the cytoplasm and go back into the SR. And so the calcium needs to be actively pumped back into the sarcoplasmic reticulum where it was being stored once again. And so when it leaves the sarcoplasm or the cytoplasm, it is then going to result in the covering up of those myosin binding sites such that even if myosin is still present, it cannot physically make contact. It cannot bind because those myosin binding sites have now been covered up. And so that is really important to generating muscle relaxation. So muscle needs to contract when it should, but it also needs to relax when it is time. This is why excess calcium can result in spastic contractions, because it is opening up those binding sites inappropriately and the muscle does not have time to relax. Okay, so just looking at these two configurations, this is it in its relaxed state where these binding sites are covered up by tropomyosin. This is if calcium is not bound to troponin. And then in its contracted state, calcium comes in, binds to troponin, drags those tropomyosin proteins off of the binding sites, and so myosin is free to come, make interaction, and start that crossbridge cycling. Okay, again, another helpful video here in visualizing that as well. Okay, here is the gating mechanism. So when the action potential runs down the T-tubules, it is going to activate the DHP receptor that is touching the ryanodine receptor. And so the action of that energy that activate that the end plate potential, coming down the T-tubule touches on the DHP receptor, that touches on the ryanodine receptor, and this opens up that channel so that calcium can leave the SR. So this area should be in blue. This is representing the SR over here. This is the SR membrane. And so we need to get that calcium from the SR where it's being stored out into the sarcoplasm so that it can go and interact with troponin and generate that, that sliding of the tropomyosin to bring about crossbridge cycling. Okay. All right, so I think that's it in terms of topics I wanted to review that are going to be really important for the exam. Any questions from muscle things? Any questions from this? So the exam will be up to excitation, contraction, coupling. So it will include the structure.